In the last lecture, we started talking about neurotransmitters and their metabolic pathways, their synthesis and degradation. And today we're going to look at how these neurotransmitters actually exert their effect on the cells. So we're going to talk about our neurotransmitters and their receptors. So we're going to be talking about receptors of neurotransmitters. We're going to be talking about their different signaling cascades, because as you will see, one neurotransmitter or one signaling molecule or one hormone, whatever you want to call them, can have different subtypes of receptors, and those subtypes of receptors can start different signaling cascades, and therefore the effect of a given molecule, of a given signaling molecule, hormone, neurotransmitter, can differ depending on which receptors are expressed on a specific cell or in a specific region or specific tissue. Okay? So one important thing to realize at this point, and it's quite possible that you've already realized it throughout the course, but if you haven't realized it now, that one molecule can have different effects in different tissues depending on the receptors that are expressed there. Okay? And also these effects can be exactly opposite, and we'll see that for some of them, we can have opposite effects of the same molecule on different receptors. Okay? So this is an important thing. When we talk about the central nervous system especially, it's even more difficult to say, well, this neurotransmitter, if its concentration or if its production increases, is going to have this effect. And that's caused, first of all, by the fact that there are different receptors in different areas of the brain or central nervous system. But also it's given by the fact that the central nervous system is a hugely complicated network of neurons and glia, as you probably know. And therefore, you know, influencing one little part of the brain or the spinal cord or what have you can have completely different effects somewhere else, and it's a hugely complicated endeavor. So when in this lecture and the last lecture, when I say, well, there is a hypothesis that an increase of this neurotransmitter probably causes such and such disease, I'm going to mention it today as well, it's a huge simplification to say that, okay? So even when I do say it, take it as a simplification, because it's impossible, in most cases it's impossible to say, okay, dopamine, you know, too much dopamine causes schizophrenia or something like that. It doesn't work like that, okay? It's a simplification. Okay. All right, to start with, what is a receptor? What is it?
Protein.
Okay, it's usually a protein. Almost always a protein. Yeah. There are lots of proteins, not all of them are receptors. What is a receptor? Yeah?
Have some extracellular domains, transmembrane domain, membrane domain.
Okay, so what you're saying is that there are integral membrane proteins. Do receptors have to be membrane proteins? No? Okay, they don't have to be membrane proteins.
Have binding sites
Binding sites.
or other ligands.
Okay, for ligands. So a receptor is usually a protein, almost always a protein, which can bind a ligand. That's all that we need for a definition of a receptor. Okay, it can be on the membrane, it could be in the cytoplasm, it could, in theory, be extracellular, it could be anywhere. It just needs to be a molecule that is able to receive, hence the name, it's a receptor, to receive a ligand, to bind a ligand. Okay, that's all we need for definition of a receptor. Now, as you know, those receptors, especially the membrane receptors, but not only those, can start various signaling cascades. Okay, so they can transfer the signal, in the case of membrane receptors, from the extracellular space to the intracellular space. There are two large groups, or we can take all the receptors, all the membrane receptors, and divide them into two huge groups, depending on the type of process that they start, that they get going in the cell. Does anyone know which two groups I'm thinking about?
Associated with G protein or?
Okay.
Or
Okay, so we have receptors associated with G proteins, correct?
ligand-gated channels?
Ligand-gated channels, okay.
Voltage-gated channels.
But those voltage-gated channels aren't really receptors, because they don't bind any ligands, do they? At least under normal conditions, obviously. You know, there are toxins that, as we talked about it, but normally they're not regulated by ligands, so they're not receptors, okay?
Intrinsic enzymatic activity?
Okay, so the large distinction that I was talking about, the two categories of receptors are ionotropic and metabotropic. Okay, and I know from the Czech group that you've probably never heard those two terms, but they're quite important. So I'm gonna. Okay, so we can take all the receptors, especially membrane receptors, but not only them, and divide them into ionotropic and metabotropic. Ionotropic receptors are ligand-gated ion channels. Okay, and ionotropic means just that they do something with ions. Okay, metabotropic, and it's a historical name, says that those receptors do something with metabolism. They change metabolic pathways or they change metabolic processes in the cell. Okay, so you can imagine that ionotropic receptors are sort of electrical. They move ions or they let ions move, while metabotropic are sort of chemical receptors. Okay, they start a chemical reaction in the cell. So out of those groups that we just mentioned, which receptors, which of those will go under ionotropic and which will go under metabotropic? The ones that you just mentioned.
G protein under metabotropic.
Okay, G protein coupled receptors, definitely metabotropic.
Ligand-gated ion channels under ionotropic.
Indeed, ligand-gated ion channels are ionotropic. That's actually a synonym because that's what they are. Okay.
Tyrosine and serotonin linked kinases is metabotropic.
Correct. Okay, so all the receptors that are associated with protein kinases or that have their own intrinsic protein kinase activity are all metabotropic. Okay, so do you see the distinction? Because I'm going to be using those terms throughout this lecture, and I think it's an important distinction. Okay. Right. Okay. Well, let's start with the first neurotransmitter system, and we're going to be talking about the receptors for catecholamines, specifically adrenaline and noradrenaline, because dopamine has different receptors. But adrenaline and noradrenaline bind to the same receptors, and we call them adrenergic receptors. Adrenergic receptors. Okay? So adrenergic receptors bind both adrenaline and noradrenaline. But the affinities, and we're going to be talking about affinities more in the next lecture, but the affinities, so the likelihood that the receptor will bind adrenaline or noradrenaline differs between the different subtypes. So I'm going to mention that as well. Adrenergic receptors are divided into two large groups, alpha receptors and beta receptors. Okay? So you hear about alpha adrenergic receptors and beta adrenergic receptors. Now, to make it even more complicated, even alpha and beta are divided further into subtypes. Okay? And we recognize alpha 1 and alpha 2 receptors, and then beta 1, beta 2, and beta 3 receptors. Okay? Now, this division into subtypes is, first of all, given by their different structure. Okay? So the receptors are actually chemically different. Okay? The proteins are different. They're very similar to each other, but different enough. But the more important reason why we divide these, adrenergic receptors into these subtypes is that, first of all, they can bind different pharmacological ligands. So we can actually find specific ligands for alpha 1 and alpha 2. So all of these bind adrenaline and noradrenaline, okay? But we can find specific chemicals that bind to alpha 1 or specific chemicals that bind to beta 2, which is obviously useful to influence them. And the third reason why it's important to divide them is that the different subtypes of adrenergic receptors have different effects on the target cells, okay? Now we're going to talk about the effects and the signaling cascades that they start. Okay. All adrenergic receptors are metabotropic, all of them, and they are all coupled with G proteins. So all of them are G protein coupled receptors. Okay? Actually, G protein coupled receptors are the largest group of receptors, so we'll see that actually most of the receptors we're going to be talking about are G protein coupled, not all of them. Okay? Yes?
You said that the adrenaline and the noradrenaline bind to all the alpha and the beta receptors, but the affinity is different, so as to say the adrenaline binds to the beta and the noradrenaline binds to the alpha receptors.
Correct. Okay, I haven't said that. I was going to, but yes, you're right. Okay, I mentioned that there are different affinities for adrenaline and noradrenaline. So noradrenaline is more likely to bind to alpha receptors than adrenaline, and adrenaline is more likely to bind to beta receptors than noradrenaline. Okay, so if we have a, does it make sense? Yeah, if we have a mixture of both adrenaline and noradrenaline, alpha receptors are more likely to bind noradrenaline and be activated by it, and beta receptors are more likely to be activated by adrenaline. Okay, but both activate both. Okay, only the affinities differ. Yep. Okay, that's actually a very useful thing to remember, especially for pharmacology, because well, I'm first going to talk about the effects of these neurotransmitters, and we can go back to the effects of adrenaline and noradrenaline that you give to a patient. Okay, right. Alpha-1 receptor is coupled to GQ protein. Now, Czech students told me you've never heard of GQ protein.
We have.
Okay, well, very good. Right, so what is the signaling cascade from GQ that is activated by GQ, by the alpha subunit? So what does it activate? Okay, activates phospholipase C, which then...
IP3 and diacylglycerol.
So...
IP3 and diacylglycerol splits into IP3 and diacylglycerol.
Phospholipase C splits into those? What is PIP2?
Phosphoinositol phosphate.
Okay, anybody can get the right name? Okay, phosphatidyl inositol bisphosphate, right? PIP2 is split into, well, give me the whole names. Inositol trisphosphate and diacylglycerol. Okay, is that the end of it? No?
IP3 goes to the smooth endoplasmic reticulum and activates HCA.
Okay, inositol trisphosphate, there are specific receptors on the endoplasmic reticulum for IP3, which open, and there are calcium channels, so they allow calcium to go out from the endoplasmic reticulum, increase calcium concentration in the cell. Very good. PKC, and you're right that calcium can activate PKC as well, because there are different subtypes of PKC. Right, okay. It depends which subtype of PKC we're talking about. Okay, I think there are about 10 different PKCs at least, and they're activated by different things.
When calcium is not released from ER, but how does it rise in smooth muscle?
Well, by the same mechanism. Oh, there is, definitely. Okay? When we talked about smooth muscle being less dependent, being more dependent on the extracellular calcium, that doesn't mean that it doesn't have any calcium in the endoplasmic reticulum. Okay? It definitely does. Okay? Substantial amount. Okay? Just needs extracellular as well. Okay?
But then isn't the phosphokinase C enough to phosphorylate the myosin light chain in smooth muscles, or do we still need the phosphorylate? PKC doesn't really phosphorylate it, or it definitely doesn't phosphorylate it at the place where it's needed for contraction. We need myosin light chain kinase for that, which is activated by calcium. Okay, so PKC, no. Right, yeah?
Sorry, which phospholipase C is this? Is it beta or gamma?
I don't know. Does it make a difference?
Yeah, I think we took that in one of the lectures.
Okay, I don't know. Honestly. Yeah, I don't know. I can find that out for you if you'd like. Okay, so this is the signaling cascade for the alpha-1 receptor. Now, alpha-1 receptor, and actually both alpha receptors, are present in the central nervous system, so they do things there. Do you think alpha-1 receptor is going to excite the cell, or is it going to inhibit the activity of the cell? Judging by the signaling cascade. It's definitely much more likely to excite it, because you can see that we're increasing intracellular calcium, which is generally a signal for the cell to do something. Okay? It's again a simplification because the whole cascade obviously is much more complicated and is influenced by other signaling cascades. But in general, you can say that yes, when you see a GQ protein, usually that's something that stimulates the cell either to produce neurotransmitters or to contract or what have you. Okay? Now, I said that both alpha-1 and alpha-2 in the central nervous system, but alpha-1 is also a very important receptor on the smooth muscles of blood vessels. Okay? So the smooth muscles of blood vessels contain alpha-1 receptor. Now, what's going to happen if we activate alpha-1 on smooth muscles? Okay, there's going to be a contraction, okay, because of the increased calcium and obviously other processes that happen there. Okay, so what happens with the blood vessel itself if the smooth muscle contracts? Vasoconstriction. Okay, it's going to be vasoconstriction, so the lumen of the blood vessel is going to get smaller. And what happens to the blood pressure? It goes up. Okay, so alpha-1 receptors, apart from being in the central nervous system, they are on the smooth muscle of blood vessels, and they mediate the effect of catecholamines to increase blood pressure, okay, to constrict the blood vessels, okay, through alpha-1. This is going to become important in the last lecture when we talk about drugs that can treat increased blood pressure, okay, so alpha-1 receptors. Right. Alpha-2 receptors are primarily in the central nervous system, so we don't really find many of those outside the central nervous system. They are coupled with a GI protein. What is the signaling cascade from GI? Okay, so it inhibits adenylate cyclase and therefore decreases CAMP. Okay? I'm going to tell you another huge simplification. A GI protein generally means an inhibitory effect, okay? It doesn't have to, but very often it does, okay? So a rule of thumb, if you see a GI protein, usually that's an inhibitory receptor, okay? Usually, not always. Okay? Now, alpha-2 receptors are, remember when we were talking about synapse, alpha-2 receptors are the presynaptic receptors for catecholamines, for adrenaline, noradrenaline, okay? Presynaptic receptors. So what is their role? Okay, so they give negative feedback to the presynaptic terminal to say, enough, okay? You've already released too much adrenaline or noradrenaline into the cleft, okay? Stop releasing it. That's why they are inhibitory, okay? All right? So the purpose of presynaptic receptors is to reduce the release of the neurotransmitter into the synapse, into the cleft. Okay, it's a negative feedback. So if there's too much neurotransmitter, it tells the presynaptic neuron stop releasing anymore. Okay, and hence they obviously have to be inhibitory because it's a negative feedback loop. Okay, does this make sense? Alpha receptors, alpha adrenergic receptors. Okay, let's have a look at the beta receptors. It's going to be slightly easier because they are all coupled to the same G protein, which is a GS protein. Okay, what is the cascade that is started by G alpha S? Okay, so it stimulates adenylate cyclase, therefore increases CMP. Now, the GS, the S stands for stimulating, okay? But in fact, while we had a nice simplification here saying that GQ is generally exciting for the cell, it usually activates the cell, and GI generally inhibits the cell. For GS, it's actually quite difficult to make this simplification because the activation of G-alpha-S can either be activating for the cell or it can inhibit its activity depending on what happens after the increase of CAMP, what kind of other proteins that are dependent on CAMP or react to CAMP are present in the cell. Okay, so here it's actually quite difficult to simplify, to give you a rule of thumb, and you're just going to have to remember which receptor does what. Okay, so beta receptors are also present in the central nervous system, but a bit less than alpha receptors. So alpha receptors are really important in the central nervous system. Beta are not as important, but they are very important in peripheral tissues. I'm going to tell you about their functions. Beta-1 receptor is present in the heart, okay, in the heart muscle, in cardiac muscle, and especially in the conductive system. And there, it mediates the effect of catecholamines on the heart. Does anyone know what adrenaline, noradrenaline does with the heart? If we produce adrenaline, noradrenaline?
Increase the heart rate.
Okay, increases heart rate.
Blood pressure as well.
Okay, yeah. Okay, yeah. So, yeah, correct. So it basically stimulates the heart, okay? It increases heart rate, it increases the strength of contraction, it also increases the speed of conduction, okay? So that the stimulus is conducted faster in the heart. You said correctly that it's going to increase the blood pressure independently on the effect on alpha-1, and here I'm getting into second-year territory, but basically, if the heart is pumping faster and more, it's obviously pumping more blood into the same circulatory system. So if you pump more, obviously the pressure is going to be higher, okay? I don't want to spend a lot of time with that, but that's the mechanism by the heart itself, without any reaction of the blood vessels, can increase blood pressure, okay? So you're right. The activation of beta-1 receptor in the heart causes increased heart rate, therefore increased blood pressure as well. Okay, so that's beta-1 receptor. And again, we're going to spend a bit more time in the last lecture talking about all the various drugs that we can use to influence that. Okay. Beta-2 receptors are expressed, I mean, at least from clinical point of view, in two important tissues. One is the smooth muscles of blood vessels, and the other one are smooth muscles of the bronchi in the lungs. Okay? But unlike alpha-1 receptors, these beta-2 receptors cause relaxation. They relax the smooth muscles. Okay? Why is that? Well, CAMP activates protein kinase A, and protein kinase A can phosphorylate myosin light chains. But it phosphorylates them at a different site than myosin light chain kinase, and this phosphorylation causes an inhibition of contraction. Does it make sense? Yeah? So myosin light chain kinase phosphorylates here, causes contraction. Protein kinase A phosphorylates here, inhibits contraction. Okay? So beta-2 receptors, through G-alpha-S and all these things, cause relaxation of smooth muscles.
Is that protein kinase A and G, do they have the same function but just in different locations?
Correct. Yeah, so protein kinase G, we're going to mention it very briefly, are we? Yes, very briefly at the end of the lecture, which is activated by, well, anyway, we're going to talk about it. But yes, it has a very similar activity, only phosphorylates a different residue.
What about PKC and PKA? The difference, don't they both relax persons?
PKC? I don't think it does. But it depends which PKC, which muscle. It's generally, that's not the case, okay? Yeah? Now, you might ask, why do we have both alpha-1 receptors on blood vessel smooth muscles and beta-2 receptors? Aren't they going to work against each other? Is that something that crossed your mind? Okay, maybe. Right. Well, there are two main differences between, yes?
No, because in some tissues I need in special reactions less blood, like in some organs, like the intestine, but otherwise I need, for example, a lot of O2 to have a reaction. That's why we increase the diameter of the bronchi, so I get.
Okay.
So,
yep.
Some tissues are not as important as others in special type.
Okay, correct. The explanation for this seeming paradox is that, first of all, think about the affinities. So adrenaline will activate different receptors, or will preferentially activate different receptors than noradrenaline. But the other, and that's the one that your colleague hinted on, is that different tissues, different types of blood vessels in different tissues have different receptors. Okay? Now, adrenaline and noradrenaline, as you probably all know, one of their functions is to regulate or to start the fight or flight reaction. Have you heard of fight or flight? Yeah? Okay, so imagine, long time ago, but obviously it can happen here as well, you're exposed to some danger and you have to either fight your way through it or you have to run away. Now, in this reaction, in this situation, your adrenal glands, your adrenal medulla is going to start pumping adrenaline or noradrenaline into the blood. Okay, what happens? Well, in some blood vessels, in some tissues, there's going to be vasoconstriction because in this situation, we don't need them. Okay, and the type of and one example of that tissue is gastrointestinal tract. Okay, we don't need to be digesting food at this reaction at this situation. Okay, we have other important things to deal with. On the other hand, blood vessels in your muscles and in the brain as well, we need to dilate those. Okay, because we're going to need as much oxygen, as much blood, as much glucose as possible, so that we can actually do what we need to do. Okay, so even though we basically have one signaling molecule, or two of them, depending on which receptors are in the specific tissue, we can have exactly opposite effects. Okay? So there's going to be vasoconstriction in one part of the body, and there's going to be vasodilation in other parts of the body, and it all makes sense because they need to work in a coordinated way so that we survive. Yep.
Is that the same reason why we can go for swimming after we had lunch?
Generally, no. I mean, first of all, it's questionable whether it's really a problem.
You have to have some. Your intestines must work during the.
Yes, I think there's just general idea that basically, if you're using oxygen for something else, it's a bit dangerous to go swimming because you might get cramps and you might drown or something. But I think it's more of a kind of a folk tale rather than a real danger. But the logic behind it is similar, but it has nothing to do with adrenaline and noradrenaline. Okay. There was another. Yep.
The amount of receptors if you're looking at the receptors in your small intestine versus your heart, is it that they don't have the same receptors, or they have a different amount of receptors?
They have a different mixture of receptors. Okay, so it's not even that your GIT has only alpha-1. There's actually a mixture of alpha-1, beta-2, probably some other ones as well. But the mixture is different, and therefore the effect is different. There was another.
What if adrenaline bind to alpha-1 receptor? Will you have the same cascade or different?
Absolutely, yeah. If adrenaline binds to alpha-1, it's the activation of alpha-1 receptor. The receptor doesn't care what binds to it. If it binds and if it activates it, it doesn't matter. Okay, it's really just about the affinities. So adrenaline is more likely to bind to beta receptors, but it will bind to alpha and do exactly the same thing as noradrenaline.
In the gastrointestinal tract, is the minority of the receptors alpha-1?
Correct. Yeah, there are fewer alpha-1 receptors. Sorry, there is majority. Yeah, there is more alpha-1 receptors than there are beta-2 receptors. Yeah.
In the brain, will the alpha-1 have the same reaction with vasoconstriction, or will it perform vasodilation?
Alpha-1 will always cause contraction and vasoconstriction. Beta-2 will always, no matter where it will cause dilation of the muscle if it is on a muscle cell.
That will constrict blood from coming into the brain.
Yeah.
So
When I said that alpha-1 and alpha-2 are in the brain, I was talking on the neurons. Okay? They are probably on the smooth muscle of blood vessels as well, but the ratio is going to be in favor of beta-2. Okay? So when I said that they're in the central nervous system, I was thinking about neurons. Okay? And that they are there for the signaling in the nervous system. Okay, any more questions?
Right,
yep.
You mentioned the PKA and how it relaxes the smooth muscle, like for vasodilation.
Yep.
I didn't quite understand what the process, like does it like phosphorylate something?
Yeah, I mean, basically, imagine that you have, this is myosin, and this is the myosin light chain, the regulatory one, okay? Now, myosin light chain kinase, imagine, phosphorylates it here, which activates contraction. Okay? That's the mechanism of contraction in smooth muscle, right? Yep. Just nod your head if that makes sense. Okay. PKA phosphorylates it here and blocks contraction because it's a different place, okay? And that's how the protein works. If it's phosphorylated here, no contraction. Even if myosin light chain kinase tries to contract it, it's not going to happen. Okay? PKG, as your colleague suggested, phosphorylates here, also blocks contraction.
So does it when it phosphorylates, does it always relax?
Yes. When PKA phosphorylates this specific myosin light chain or PKG, it will relax. It will block contraction. Yes. Obviously, then there are phosphatases that remove it so that we can stop the signal from. Yep. Okay. The last of the beta receptors that I haven't talked about is beta 3 receptor, and that is found specifically, or more or less specifically, on adipose tissue, on adipose cells, fat cells. Okay. The purpose of beta 3 is when they're stimulated, they increase the activity of lipolysis, okay, of breaking down lipids into fatty acids so that they can be used by the body, okay? So beta 3 receptors, adipose tissue, increase lipolysis. You've heard of hormone-sensitive lipase, okay? This is how hormone-sensitive lipase is activated. Beta 3 receptors activated, CAMP increases, which activates the hormone-sensitive lipase. Okay? Yep.
The beta 1 receptor, if you just talked about how the beta receptors cause more constriction, how cause more dilation,
yep.
sorry, why does it increase the heart rate then? It doesn't acquire the beta gamma or something, or?
No. Okay. It causes relaxation in smooth muscles, but not in cardiac muscle. Okay. In cardiac muscle, actually via CAMP and PKA, it actually increases the activity of ion channels so that the stimulation is more active. Okay. We don't quite have time to go through all the details, but the mechanism is different. Okay. It's a very different mechanism. That's why I was trying to say be careful about GS. While we can say that GQ is mostly activating, GI mostly inactivating or inhibiting, for GS that's always complicated. It depends on which target proteins are in the cell and how they are affected by CAMP and PKA and some other cascades. Okay, so it's not easy to find parallels between different tissues. Okay, right. If there are no more questions about adrenergic receptors, we're going to move on to the next catecholamine, which is dopamine. Okay, that's the one that we haven't talked about. We're going to talk about dopamine receptors. Okay, dopamine receptors are, again, all metabotropic. All of them are metabotropic. All of them are coupled with G proteins. Okay, so once again, all of them G protein coupled receptors. There are approximately, at this point, there are about 5 subtypes of dopamine receptors. I'm just going to mention the first 3 of them. No, maybe just the first 2 of them, actually. Right. Before we talk about the specific receptors, it's quite important, I think, to say the function of dopamine. What does it actually do in the brain? Okay. Dopamine is also secreted by the adrenal medulla to some extent, and there are some dopamine receptors in the periphery as well, but they're not as important as adrenaline and noradrenaline, so we're not going to talk about them. We're just going to talk about dopamine in the brain. And what is the function of dopamine in the brain? Here, I'm just going to refer to the beginning of the lecture when I said, well, it's actually very hard to say what one neurotransmitter does in the brain because it depends where it does it, when it does it, etc. So for dopamine, we recognize 4 different pathways, dopaminergic pathways in the brain, and each of them has a different function. Now, what do I mean by dopaminergic pathway? Well, I mean that there is a small group, usually quite a small group of neurons that are dopaminergic, they produce dopamine, that project their axons into some other part of the brain. That's a dopaminergic pathway. Okay? Some neurons projecting to some other part of the brain. Yep, okay. So now I'm going to tell you about the 4 different systems because they have different functions. I'm going to be using names of brain structures that you've probably never heard of, well, almost definitely. But I'm going to try to explain at least a little bit. You'll know more about them next year. But I'll try to explain at least a little bit where they are in the brain and why they're important. Okay? So, there are 2 main dopaminergic nuclei, groups of cells, groups of neurons. Okay? It's called a nucleus in the brain. So, 2 main dopaminergic nuclei in the brain. The first one is called substantia nigra. And you may have come across with it in Latin, maybe, substantia nigra. Does anyone know what it means? Black substance, okay? It just means black substance because it actually is. It's pigmented, so it actually, if you cut the brain, you see a little tiny black thing. That's substantia nigra, okay? And substantia nigra, in fact, is a small group of dopaminergic neurons that project to other parts of the brain. The other nucleus, which is also dopaminergic, is called VTA, which stands for ventral tegmental area. You don't have to remember those at this point, okay? I'm just trying to tell you about the different system. So these are the two main groups of neurons. They are all sort of in the middle of the brain, not in the end part of the brain. They're much lower down, okay? They're much older. And as I said, they project to various parts of the brain. Right, so let's...
Produce neurons that produce dopamine, or they receive dopamine?
No, those are dopaminergic nuclei, so those are groups of neurons that produce dopamine, okay, and project elsewhere, okay? Right, so since they are in the middle brain, which is mesencephalon, yeah, okay? So the first projection, dopaminergic projection, is called mesocortical. Mesocortical. It goes from the mesencephalon, okay? Actually, it's from the VTA into the cortex. Does this make sense? Yeah? From somewhere deep down there into the cortex. Mesocortical projection. What is the function of the mesocortical dopaminergic projection? Well, it looks like it's very important for correct thinking, for correct perception of reality, for understanding what is real and what is not real. Okay? It's also important for appropriate planning of your actions, understanding the social context of what's going on. So it's actually very important for normal functioning of humans in society. Okay? This pathway is one of the pathways that are damaged in, it's actually overactive, in schizophrenia. Okay? So a damage to this mesocortical pathway, which actually projects to the frontal lobes and prefrontal areas, causes quite deep disturbances in the perception of reality, in functioning in normal society. Okay? So that's the mesocortical pathway, mesocortical projection. The other one that I want to mention is called mesolimbic. So once again starts in the mesencephalon, the VTA, and it projects into what we call the limbic system. The limbic system, I'm not going to go into details, but it's responsible for the regulation, for the function of emotions, emotional reactions, aggression, all sorts of things, okay, sexual drive, etc. The mesolimbic dopaminergic projection is a very important, is a central part of something that we call the reward system. And as the name suggests, it's a neurological and neural circuit that provides the brain with reward. Okay, so if we do something that the brain thinks is good for us, we get the feeling that it's good. Okay, so that's what the reward system is for. Okay, so if we eat a good meal, the reward system starts working and says, okay, that was good. Okay, if we run a marathon, it tells you, oh yeah, great. Okay, if you have sex, the reward system tells you, yeah, keep doing that. Okay. So the mesolimbic projection, which is dopaminergic, it's dependent on dopamine, is part of the reward system. Why is it important clinically? Well, most importantly, the reward system is very active, is very important for the development of addiction. Okay, which makes sense because addiction is just basically doing what gives you the reward. Okay, and quite a few drugs, and we're going to talk about them in the last lecture. I mean, illegal drugs, drugs that are abused, activate this reward system. Some of them directly, and some of them indirectly, and that's the reason why they become addictive. At least part of the reason why they become addictive. Okay. Heroin? Heroin. Yeah, heroin actually activates it indirectly, but yes, cocaine activates it very directly. Okay. Right. So that's the next projection. The third projection, do you have a question about this specifically, mesolimbic?
Which one produces the one, like dopamine 1 and dopamine?
Yeah, I'm not going to speak too much about the specific distribution of these two receptors, okay? Because it's actually quite complex. I mean, D2 receptors are definitely present in the mesolimbic system, to some extent in the mesocortical, and definitely in the third one that I'm going to be talking about. The D1 receptors are all over the place, but it would be way too complicated to talk about the individual distributions, okay? So I'm not going to talk about that. Right. The third system that I want to talk about, and here we're moving from the VTA to the next nucleus, which is substantia nigra. So that's called nigrostriatal. Nigrostriatal. Okay, can you read it? This is STRI. Okay, so this projection goes from the substantia nigra we talked about and goes into another structure which is called corpus striatum, a striated body, okay, which is another nucleus in the brain. The corpus striatum is part of a very important structure called basal ganglia. And basal ganglia, very large nuclei at the bottom of the brain. They are very important for the proper execution of stereotypical movements. So proper, walking, keeping your posture, to some extent writing and typing, and all those things that are kind of repetitive. You don't really think about them. They are executed to a large part by basal ganglia. Okay. This nigrostriatal pathway plays many roles, but one of the roles that it plays is that it allows the initiation of movement. Okay? So if you want to start moving, you need the activity in the nigrostriatal pathway. Why is that important clinically? Well, nigrostriatal pathway and substantia nigra are the structures that are damaged in Parkinson's disease. Okay? So this is the dopaminergic pathway that is damaged in Parkinson's disease, and this is what causes the typical symptoms, so inability to initiate movement. Okay? Very typically, the patients, it's very difficult for them to start moving, okay, to lift their legs properly. Their face expressions are very slow, and usually they don't have much, when you see them speak, they don't actually change their facial expression at all. It's also caused by a dysfunctional basal ganglia due to damage to the nigrostriatal pathway. Yep?
Would alcohol, does it do anything with this part?
Not really, no. Why are you asking?
No, I'm just wondering because when you're drunk walking.
Yeah, I mean, for alcohol, it's actually much more, it damages the function of the cerebellum, which is also important for coordination. So these pathways work together in a way. Basal ganglia, all the structures, and cerebellum, they all are important for different aspects of the movement. But I'm stressing that it's the initiation of the movement, okay? Cerebellum makes sure that you coordinate it properly, okay? In time, in space, etc. Right. The last projection I want to mention before the break is called Tuberoinfundibular. The name is a bit strange. It doesn't really originate in either of those two nuclei. There's another nucleus. I'm just going to mention I'm just going to talk about the function. It's present, this pathway, Tuberoinfundibular. Okay. It's in the hypothalamus, so somewhere behind there, okay, in the hypothalamus. And this pathway is the one that blocks the production of prolactin. Okay. So you talked about prolactin. Okay, so you know its function. And you know that dopamine is the inhibitory factor, the prolactin inhibitory factor. Yeah? And this dopamine comes through the tuberoinfundibular pathway. So that's the fourth pathway that is important, again, clinically, because when we talk about drugs that block dopaminergic transmission, we'll see that it causes problems with basically all of those. Okay, so tuberoinfundibular blocks the production of prolactin. If we block it, we get high production of prolactin. Yeah.
Would you like us to know these pathways for the exam?
Yes.
So is it the dopamine that deactivates this pathway which causes the, like, is it dopamine that deactivates it, or?
Which one?
The thing that you just talked about.
Okay, no. This pathway is the dopaminergic pathway, okay? So it's the pathway through which a group of neurons secretes dopamine into the hypophysis, into the pituitary gland. Okay? So if it's working, it blocks the production of prolactin. If it's not working, prolactin is going to be produced in an uncontrolled way.
So is it the dopamine that deactivates the prolactin?
Yes, absolutely. Yeah, it's the prolactin inhibitory factor.
But these make the dopamine?
Okay, I'll try to maybe draw a picture. Okay, so what do I mean by these projections? So if this is the brain, okay, something like that. Okay, let's say that this is VTA or thereabouts. Okay, a tiny little group of neurons. Okay, and it projects into the cortex. Okay, projects into the striatum somewhere here. Substantia nigra, sorry, yeah, the projection. Substantia nigra, here it goes into nucleus accumbens, and here it goes into the pituitary gland. Okay, so these are just axons, basically, of neurons that produce dopamine in the target. Okay, I can see that you're really confused, so I didn't really explain it well, I guess. These pathways are just axons of dopaminergic neurons that go to a target structure, and there they release dopamine. That's what they are. Okay, so there are neural circuits, there are neural pathways that use dopamine as the neurotransmitter. That's all they are. Okay, and they start with where the nerve bodies are, which are in the nuclei, and they end in the target structure, such as the cortex, limbic system, striatum, or the pituitary gland.
So it's not like any other neurotransmitters where it's formed at the end of the synapse?
Oh, it absolutely is. It's just that the neuron is one thing, okay? So we still call it dopaminergic neuron, even though dopamine is produced somewhere far away. So the dopamine is actually produced here, okay? But the neurons are here. So if you kill them here, you don't get any dopamine here. Yeah?
Is it like an agonist or antagonist?
What is?
Dopamine.
Dopamine is definitely an agonist, okay? Because they are dopamine receptors. The same way as adrenaline is the agonist on adrenergic receptors, etc., etc. They're always, those are the physiological agonists. Okay? Yep.
So the nuclei sends out the axons to these different...
Yes.
But is the VTA and the other one, is that the nuclear region, and then you have 4 different pathways after that?
Yes.
Okay.
Yeah.
So the VTA applies to the first...
Yeah, this goes from the VTA. This goes from substantia nigra. There's actually a separate nucleus for this one. Yeah.
Is that the reward system, is that a good thing?
Oh, yes. Well, yes, but absolutely, because it actually keeps us alive. It gives us motivation to do a lot of things.
That also stimulates addiction to some things?
It can do, yeah. That's the, that's the disadvantage of it, but it's very important, otherwise you wouldn't eat. If you have a rat and you destroy the reward system, it dies. It doesn't drink, it doesn't eat, because it doesn't get the reward for it.
Yeah, but I think there are some people that are addicted to dopamine.
Yeah, yes,
So
ish, yeah.
That's not like opposite things, like the reward system and the...
Well, how to say it? I mean, if you have hypertension, the hypertension in part is caused by your blood vessels and your heart. But you can't say, therefore the heart and the blood vessels are a bad thing. No, they're not. I mean, you need them. Yeah, sometimes they can cause hypertension. Okay? So the reward system is absolutely essential for survival. But sometimes it causes addiction, yes. I'm not sure if I got your question, but... Okay, just very quickly, and we'll have a break. We're very, very far behind. Let me just mention that D1 is coupled with a GS protein, and this one, D2, with GI protein. D1 generally is more a stimulatory receptor, and D2 is more of an inhibitory receptor. Okay? Okay, let's have a 3-minute break. Okay? I will ask whether you have any questions, but I think we had plenty of questions during the lecture, so unless there is a really pressing question at this point, I'm going to move on to the next neurotransmitter system, which is serotonin. Okay, so we're going to talk about serotonin and serotonin receptors. Now, serotonin receptors are usually labeled as 5-HT receptors. Okay, so 5-HT is the usual abbreviation. In some older literature, you can sometimes still find S, but that's not the standard way of labeling them. So 5-HT are the receptors. Once again, we have a lot of subtypes. At this point, I think there are at least 5 subtypes of 5-HT receptors. And those subtypes, to make it even more difficult, have subtypes. Okay? But I'm just going to talk about 3 of them. Okay? And I'll try to explain why I talk about these 3. Before I do, well, actually, no. Let's talk about them first. Okay. So far, all the receptors have been metabotropic, and all of them were coupled with G proteins. Now we get the first exception. 5-HT3 receptor is an ionotropic receptor. It's a ligand-gated ion channel. And in this case, it's a non-specific cation channel. What does it mean, non-specific cation channel? It just means that when it opens, it allows cations to go through. What cations can go through? Sodium, potassium, calcium. Okay, those are the main ones. Okay, all the other ones are minority. Okay, so the opening of non-specific cation channels almost always causes depolarization. Because even though, so sodium will be coming in, calcium will be coming in, potassium will be going out, okay? But the flow of positive ions that go in is usually more prominent, and therefore the opening of cation channel causes depolarization. So those are definitely activating. There are excitatory receptors, okay? 5-HT3. Ligand-gated, non-specific cation channels. What is their function? What do they do when they activate? Well, the 5-HT3 serotonergic system has a very specific role in the central nervous system because it regulates vomiting, okay? And a very specific kind of vomiting. The brain has a very small part which all the time monitors the composition of the blood. Okay, it's called area postrema. It's a detector, it's a special chemical sensor that detects the composition of the blood. And when it finds out that there is something toxic in the blood, and the toxins can be quite broad, it causes vomiting. Okay? The logic behind it is I think makes sense. If you eat something bad, the brain notices and basically makes you remove at least some of it, which is still in the stomach. Okay? So it's a specific type of vomiting, okay, which is caused by some bad chemical in the blood. And this is mediated by 5-HT3 receptors, okay? There is obviously a whole neural system around it, but we don't need to go into details. Why do I keep talking about it? Well, it is quite important, especially for cancer treatment, okay? For a long time, because you know that most cancer treatments, and we're going to talk about them later in one of the later courses, most cancer treatments are extremely toxic, okay? That's the point, because we're trying to kill the cancer cells, okay? So they're very toxic, and the brain notices them and causes really intractable, really, really bad, really severe vomiting. And you know, in the past, this vomiting was very often the reason why we had to stop the treatment, because the patient just couldn't take it anymore. Okay, imagine that you're vomiting for 3 days, non-stop. That's just horrible. So this vomiting actually was the thing that limited the use of some certain cancer treatments. Fortunately, quite recently, I don't know, maybe 20 years ago, specific inhibitors, specific antagonists of 5-HT3 receptors were developed, which are extremely effective anti-emetic agents. They stop this kind of vomiting. And this actually caused a small revolution in cancer treatment, because suddenly we can give much higher doses to those patients, and either they don't vomit at all, or they just feel nauseous or vomit at least just a little bit, okay? But not the massive vomiting that used to be limiting the biggest problem in the treatment, okay? So this is the reason why I want to mention 5-HT3 receptors, very important for clinical practice, especially in oncology, okay? But once again, there are different kinds of vomiting. If you stick your finger in your throat, 5-HT3 receptors will have no effect. Okay, that's a completely different mechanism of vomiting. Okay, just to make it clear. Okay, so for chemical vomiting, right? 5-HT2 receptors are, I mentioned them because they have several subtypes, A, B, C, maybe even D, which is not something that you need to remember. But the interesting thing is that 5-HT2A receptors are the target for several hallucinogens, especially LSD. LSD binds to 5-HT2A receptors. Okay? So these receptors clearly have something to do with the perception, especially visual perception, of reality and understanding, once again, putting together a real world as opposed to some unreal worlds, okay? So again, this is kind of pharmacologically interesting receptor. It is clear that even in the absence of LSD or mescaline or what have you, these receptors probably do the same thing in the brain. It's just easier to study them using hallucinogens rather than in any other way, okay? And 5-HT1 receptors are there so that we have the first 3 receptors, okay? The G proteins for the first 2 are like so. The first one is GI. So 5-HT1 receptor is coupled with GI protein, and 5-HT2 receptors are with GQ proteins. So once again, the rule of thumb, 5-HT1 is generally the inhibitory receptor for serotonin, and 5-HT2 is generally the excitatory receptor for serotonin. The other ones we're not going to mention at this point. Okay? Talking about brain structures, there are once again specific, very small nuclei that contain neurons that secrete serotonin. They are called nuclei raphe. You don't have to remember that. Okay, I'm just putting it down if anybody's really interested. Nuclei raphe. They are again very, very deep in the brain. Okay, they're not in the cortex or anywhere near there. And they project to actually all sorts of places. They project everywhere. The function of serotonin in the brain, apart from the ones that I mentioned, serotonin is very important for keeping the right mood. Okay, so it regulates mood. Okay, whether you're happy or sad, serotonin plays an important role. It's not the only one, the only system that does it. The serotonergic system is very important for that, and we're going to mention it in the last lecture because we're going to be talking about mood disorders and how they are treated. Serotonergic systems are also important for pain regulation, okay? So serotonin can decrease pain perception in specific areas of the brain. Okay, yep?
Are there any receptors that regulate, like receptors for pain more than others? Like inhibit pain more than others?
Say again, are there any receptors?
Receptors like 5-HT1, HT2?
Oh, not that I know of. I mean, once again, there are 2 different ways of describing the function of neurotransmitters. One is sort of pharmacological, and that's the one that I use, because we know that these compounds block 5-HT3 receptors because we can discover that in the lab, and then we can say, okay, so if it blocks vomiting, therefore 5-HT3 receptors do something with vomiting, okay? Same for the other ones. The other way of describing the function of a neurotransmitter is by looking at the neural pathways that are in the brain. So there are definitely serotonergic neural pathways that activate or rather decrease pain perception. But pinpointing the specific receptor might be a lot more difficult in this case than just saying that, 5-HT2A binds LSD. I'm not sure if that answers your question, but I'm just going to have to leave it at that. Okay? If there are no more questions to serotonin, once again, we're going to talk about it a bit more in the last lecture. We're going to move on to the next neurotransmitter, which is histamine. And you'll see that we talk in less and less detail as we go further in those other neurotransmitters, okay? So this was probably the most, adrenergic and dopaminergic was the most detailed. We're going to talk less and less about the other ones. So the next one I want to mention is histamine. And we have 3 so far described receptors, subtypes for histamine. Okay. They are all metabotropic. They are all coupled with G proteins. Okay, and the G proteins are GQ, GS, and GI. You know the rule of thumb. I'm going to start with the functions. I'm going to start from the back, from H3 receptor. H3 receptor is an autoreceptor. It's a presynaptic receptor. So the same way as alpha-2 provides a negative, the negative feedback, H3 is the same thing. It's an autoreceptor. It's a presynaptic receptor. Okay. GI protein, that makes sense because it needs to be inhibitory. Okay. H2 receptor is primarily expressed outside the brain. Okay, so it's not really a neural receptor. It's a peripheral transmitter receptor, and it has a very specific function because it is expressed in the stomach epithelium, and there it regulates the production of acid in the stomach. Okay. Once again, this is very important for pharmacology because we can use H2 receptors, and agonists and antagonists, to regulate how much acid is produced in the body, in the stomach rather. Okay, so for treatment of gastric ulcers. Now, H1 is expressed in the brain, and there the roles of the histaminergic system in the brain are much less studied than the other ones, but it's clear that it regulates sleep and appetite. Okay, so histaminergic system, sleep and appetite are the main roles. Okay. Any questions?
Can you repeat that?
Which part?
About appetite and everything.
Okay, H1 receptors in the brain. The histaminergic system, which works with H1 receptors, regulates sleep and regulates appetite. That's all you need to know at this point. Okay. Right. Moving on. Yes. H2 receptors stimulate. They stimulate the production of acid, of hydrochloric acid. Okay.
What about H3?
H3 are autoreceptors, so they are presynaptic receptors for histamine. Okay. So let's move on to cholinergic receptors, to acetylcholine receptors. Right. Acetylcholine receptors, yes?
Yeah, something about the H1.
Yes?
You said that it's found in the brain.
Yes?
So if we have like an effect of those receptors, we have to take some drugs.
As far as I know, there is no condition that would be caused by a defect in H1 receptors. Okay? They are important in clinical practice because H1 receptors are not just in the brain, they're also on the immune cells. As you probably know, that histamine is a mediator of inflammation and allergic reaction. Okay? This is all mediated by H1 receptors as well. So we have H1 receptors in the brain, and we have H1 receptors on immune cells. Now, if we want to block an allergic reaction, we want to block H1 receptors. But the problem is we don't only block them on immune cells, we also block them in the brain. And this is why many antihistamines are sedative, they cause sleep, because they're interfering with the histaminergic system in the brain. So that's why they're important. They're actually much more important because they're influenced by drugs that actually want to do a completely different thing. Does that make sense?
Yeah, but how can they be like blocked if they are like those that are in the brain? Can the drugs cross the blood-brain barrier?
Well, some of them can, and that's the problem. But the newest generation of antihistamines don't cross the blood-brain barrier, and therefore they're not. They don't induce sleepiness or anything like that. Yep.
You answered my question.
Okay. Right. Let's go to acetylcholine receptors. There are two large groups, or two basic groups of acetylcholine receptors. One group are ionotropic, and the other one are metabotropic. The ionotropic receptors are labeled as nicotinic. Nicotinic receptors, and the abbreviation is NHCR. Nicotinic acetylcholine receptors. And the reason why they're labeled nicotinic is that nicotine activates them. Okay? So nicotine is a pharmacological agonist. The physiological agonist is acetylcholine. The pharmacological agonist, one of them anyway, is nicotine. Okay? That's why they're called nicotinic. The other group are metabotropic. And they are called, once again, by their pharmacological agonist, and they're called muscarinic. Muscarinic. Muscarine, does anyone know where that comes from? Yeah, okay. The fly agaric or whatever you call it. Yeah, from kind of the red toadstool mushroom. Yeah, muscarine. Anyway, so they're called muscarinic, and the abbreviation is M, right? There are several subtypes of nicotinic acetylcholine receptors. We don't really need to go into those details. I'll just say that they are relatively non-specific cation channels, okay? So once again, they let in mostly sodium, but they also probably let in some calcium and maybe some potassium out, okay? But as with most of these non-specific cation channels, they let in mostly sodium, okay? So they're definitely depolarizing, and we find them, first of all, on the neuromuscular junction, okay? Neuromuscular junction, nicotinic acetylcholine receptors are absolutely, those receptors that are there, okay, the only ones. Okay, now you look at me like that's well-known facts that everybody knows, or that you're confused. Which one is it? Okay, just raise your hand if that makes perfect sense. Not really. Okay, neuromuscular junction, do you all know what it is? Okay, neuromuscular junction, the neurotransmitter there is acetylcholine, right? Yeah? Okay, that's the only neurotransmitter there is. If we want to move a muscle, we need acetylcholine for that. Okay, and those receptors that react to acetylcholine are nicotinic acetylcholine receptors, not muscarinic. They're nicotinic. Those subtypes are on the neuromuscular junction. Okay, which is very important once again for pharmacology, because when we talk about antagonists of nicotinic acetylcholine receptors, you're going to see what they do. If we block nicotinic acetylcholine receptors, we stop moving. Okay, we're completely paralyzed. Very important. Okay, so neuromuscular junction nicotinic acetylcholine receptors. They are also present in the brain, and they are also present in autonomous ganglia, which is something we don't really have time to go into, so you don't have to remember that. Okay, but you'll see next year what autonomic ganglia are and why it's important that acetylcholine is the neurotransmitter via nicotinic acetylcholine receptors. Okay, right. There are several subtypes of muscarinic receptors, and I'm going to tell you about the first three of them, because they have different functions and different localizations, and also different signaling cascades. So all muscarinic receptors are coupled with G proteins. They are metabotropic, and they are G protein coupled with different G proteins. Okay, M1 is coupled with a GQ protein, so it's generally activating, and it's found mostly on neurons. Okay, so it's a neuronal receptor. the functions of both nicotinic receptors and muscarinic receptors, specifically M1 in the brain, there are many of them, but a very important part of their function is the formation and sustaining of memory. Okay? So even though many other neurotransmitters and many other neural systems play a role there, these, both nicotinic and M1 muscarinic receptors, are very important for the formation of memory. If those systems are damaged, that's what we see in Alzheimer's disease. So basically Alzheimer's, one of the symptoms of Alzheimer's disease is damage to these receptors, or rather the neurons that have these receptors. And there is a problem with formation of memory. Is everyone okay? Yeah, okay.
Has nicotine something to do with memory?
That's a very controversial question. For smokers, yes. So if you're a smoker, if you don't smoke, your memory is impaired. For normal people, not so much. For non-smokers, sorry. For non-smokers, it doesn't. I really didn't mean to say that, but for non-smokers, it has very little effect. So it doesn't really improve your memory. If you're a non-smoker and smoke, it doesn't improve your memory. If you are a smoker, you need to smoke to get your memory back to normal, basically. Okay, right. M2 receptors are expressed in many tissues, but one important place where they're expressed is the heart. Okay, and specifically the regulatory parts of the heart, the sinoatrial node is so the pacemakers and the conductive system. It is coupled with a GI protein, and its function is to counteract, in a way, physiologically, to counteract the effect of catecholamines. So catecholamines increase the heart rate, increase the strength of contraction. Acetylcholine via M2 receptors decreases the heart rate, decreases the strength of contraction, decreases the rate of conduction. Okay, so the opposite effect. Now, the interesting thing about M2 receptor coupled with the GI protein is that a very large part of the signaling is actually not done by the alpha subunit of the G protein. Okay, usually it's the alpha subunit that activates adenylate cyclase or what have you. Here, a large part of the signaling is done by the beta gamma dimer. Okay, remember G protein, when it's activated, splits into alpha subunit and beta gamma. In most, and probably when you had lectures with Professor Kovács, he would say, well, beta gamma just sits there or just whatever. Actually, beta gamma subunit has always some signaling, almost always has some signaling role. Okay, so it usually goes somewhere and binds and does something. Here, it actually directly influences ion channels. Okay, it activates potassium channels. So it goes on, binds to a potassium channel, opens them, and therefore, if potassium channels are open, the cells are much less likely to be excited because they're hyperpolarized. Yeah, we talked about that. Yeah, the effect of opening potassium channels. Okay, and therefore, in the case of the heart pacemaker and the conductive system, it means that the heart rate goes down, it slows down. It's less likely for the cells to be activated. Okay, super important. Okay. So that's via M2 receptors. M3 receptors are once again coupled with GQ protein, so it's a stimulatory receptor. And M3 receptors are found in exocrine glands. Okay? Tear glands, salivary glands, all those glands have M3 receptor, which activates them to start producing tears and saliva, etc. Okay? Now, why is it important? For many reasons, usually pharmacological. If you eat the fly agaric or the mushroom that contains muscarine, you will get symptoms that will be caused by stimulating these receptors. Okay? For M1, we don't really need to talk about that very much. But M2, the stimulation of M2 receptors will cause what if we eat muscarine? The heart rate will go down. You'll have bradycardia. Okay, it's a typical symptom of the poisoning. And M3 will cause Yeah, sweating to some extent, yeah. Tears, saliva, a lot of saliva being produced. Okay, so these are typical symptoms. The antidote and the antagonist of all the muscarinic receptors is another natural compound which you may have heard about, atropine. Has anyone heard of atropine? Yeah? It's a natural compound. It blocks it's the antagonist on all the muscarinic receptors, and in this case it can be used as an antidote to muscarine poisoning. Okay?
Is it an antagonist?
It's an antagonist on those receptors, yes. Pharmacological antagonist, obviously. Okay, any questions about acetylcholine? So let's move on very quickly to glutamate receptors. And the same way as we had ionotropic receptors and metabotropic receptors for acetylcholine, the same thing, the same division can be done for glutamate receptors. Okay, we have ionotropic glutamate receptors and metabotropic glutamate receptors. The ionotropic glutamate receptors, same thing as nicotinic acetylcholine receptors, they are cation channels. Okay, so when they open, they let cations in and the cell depolarizes. Okay, now their names are unfortunately once again based on specific agonists. And these agonists are synthetic compounds that when the researchers were studying these receptors, they just used various compounds and noticed which one activates which subtype of receptors, and they just named the receptors using those weird compounds that they used initially. So they're a bit strange, okay? The first subtype of ionotropic glutamate receptors, and that's actually probably the major neurotransmitter receptor in the brain. So, and there are lots of NMDA receptors. It's called NMDA, which is the abbreviation for the specific agonist, which you don't need to know. Do you? Well, maybe you do. I don't know. Look in the table. Maybe you do need to know it. It's called N-methyl D-aspartate. It's a specific agonist on NMDA receptors. Okay. The other subtype are called AMPA, which is a compound that has even more complicated names, so I'm not going to tell you even that. And the third subtype that I want you to know are called kainate receptors. Okay? Kainic acid is the specific agonist for those ones. All three of them are cation channels. They work the same way. They just depolarize the cell. Yes?
Are all three non-specific that I understand?
They are fairly non-specific, yes. Okay? Now, there's one interesting thing about the NMDA receptor. NMDA receptor is one of the best studied receptors in the brain because, as I said, it's actually responsible for probably 99% of things that go on in the brain. So it's very, very important. The interesting thing about the NMDA receptor is that at a resting potential, okay, so imagine that it's closed, the resting potential when there is no glutamate around, the ion pore is blocked by magnesium. Okay, so magnesium is sitting in the pore and blocks it. Okay, now imagine that glutamate comes in, binds to the receptor, and the receptor obviously responds to that because it's a ligand-gated ion channel. It responds to that by opening the gate and hopefully opening the pore. But magnesium is still sitting there. So in order to open an NMDA receptor, you need to have two different signals. One signal is glutamate, and the other signal is depolarization. So the membrane already has to be depolarized to some extent for the magnesium to go out and allow the full opening of the receptor. Does it make sense? So at resting potential, even though we have glutamate around, NMDA receptor is not open. We need first to have the membrane depolarized at least a little bit, and then, if glutamate is present, the ion channel actually opens and activates the cell afterwards. Okay? Before I answer your question, the purpose, or at least this shows that not only neurons are not just on-off switches, but even some ion channels or even some receptors themselves are not just simple on-off switch. And actually you can see that it does some very simple processing of the information, because it only opens when two different signals come to the same cell. Not just one, but two of them. So even at the level of this receptor, there is some processing of information. Does it make sense? Yeah? It's something that biophysicists, for example, call a coincidence detector. It detects coincidence of two different signals. Okay? So it actually is a very tiny computer, you can imagine, which only opens when two different things happen at the same time, or at least very close to each other. Okay? Very important for the formation of memory, once again, okay, for learning. Because the coincidence usually means that there are two signals coming from two different areas, and since they come always at the same time, maybe we should learn that those, or the brain should learn that those two things are associated somehow. Okay? Super important. There was a question. Yeah.
Yeah. Doesn't it, like by the time that it depolarizes and this opens, doesn't it repolarize again?
The depolarization that I talked about is not an action potential. Okay? We are not on the axon, we are on the dendrite. It's not an action potential, okay? It's just a depolarization. Remember, the non-specific depolarization that is caused by some neurotransmitter. So you can imagine that basically you have two different synapses close to each other, okay? One of them releases, I don't know, acetylcholine, depolarizes the membrane. The other one releases glutamate, and only when the two fire at the same time, the NMDA receptors open, and therefore cause proper depolarization and then an action potential later on. Yeah, NMDA receptors, very important. Right, the metabotropic receptors, there is a lot of them. They are labeled as mGlur, metabotropic glutamate receptors, many of them. I'm not going to talk about their many functions. They are in the brain, they're elsewhere. But one type of metabotropic glutamate receptor is also present in the taste buds on your tongues. And it's there to detect, well, what else? Glutamate, okay? So you've probably heard of the fifth taste. What is it? Does anyone know? Umami, yeah. So that's the fifth taste, which one of the most important chemicals that elicits this taste is glutamate, and in fact it's mediated by a metabotropic glutamate receptor on the tongue. Okay? So this is just an interesting function for one of them. But there are many, 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 many of them in the brain. They do all sorts of things. Right. Okay, super briefly. GABA receptors. There are two subtypes of GABA receptors. GABA A is an ionotropic receptor, but unlike all the ionotropic receptors that we talked about, which were cation channels, this one is an anion channel. It's a chloride channel. So GABA A is a ligand-gated ion channel which allows chloride to go from the outside to the inside of the cell, because that's the usual concentration gradient. Okay, not always, but usually we have more chloride outside than inside. So when GABA A opens, chloride goes in, and what does it do with the cell? It hyperpolarizes the cell, okay? So GABA A, inhibitory receptor. GABA B is a metabotropic receptor. It's a G protein coupled receptor. And it's coupled with a GI protein, so it's also inhibitory, as you would expect from a completely inhibitory neurotransmitter. Okay? Last group that I'm going to talk about are opioid receptors. Opioid receptors, obviously, they were first discovered because they bound opiates like morphine and heroin and what have you. But in fact, they are actually receptors for endogenous opioids. And we talked yesterday about one of them. Yeah, beta endorphin or endorphins in general. So those are the real physiological agonists for them. There are three opioid receptors, mu, delta, and kappa. So they're labeled by Greek letters, mu, delta, and kappa. okay? They are all coupled with G proteins, so they're all metabotropic. They're all coupled with GI protein. They have many roles in the brain. Obviously, one of them is regulation of pain, but not only. Their activation can cause nausea and vomiting. With some of them, they can cause addiction, depending on which receptor they activate. It's a fairly complicated system, this opiate system. Okay, but we're not going to talk about it anymore. The very last thing I just want to talk about, we talked about nitric oxide as a neurotransmitter. What is the receptor for nitric oxide? Okay, it's a soluble guanylate cyclase. And by soluble, we mean that it's not attached to a membrane. It's just sitting in the cytoplasm, okay? So in the beginning, when there was a suggestion that a receptor has to have an extracellular domain and an intracellular domain has to cross the membrane, well, it doesn't. The soluble guanylate cyclase is just an enzyme that sits in the cytoplasm. It has nothing to do with the membrane. And when nitric oxide comes along, it binds to guanylate cyclase, and guanylate cyclase starts doing its job, which is converting GTP to cGMP. And then there's the activation protein kinase G that we talked about and relaxation of smooth muscle. Okay, any pressing questions at this point? I'm really sorry that I was so slow today. Yeah?
Was this where, was it also phosphorylation of the myosin complex?
Correct, of the myosin light chain. Yes, PKG, as I showed before, yeah.
But is that for which muscle is the...
Oh, that's in the smooth muscle. Obviously, it does all sorts of other things in the brain, in the neurons. It phosphorylates all sorts of ion channels and other proteins, so the effects are much more complicated. But when talking about smooth muscle, it's the phosphorylation of the myosin light chain. There was another question somewhere? No? Okay. Yes?
I was just wondering about the GABA receptor, if it actually polarizes the cell. Because of what's the electrochemical gradient control?
Yeah, under normal conditions, there's more chloride outside. Okay, so they do really go in. It's not a massive gradient, but there is some.
But
okay.
Since the cell is negative inside one, it's not.
Well, yeah, but since there is, once again, you have an electric potential and you have an electric gradient and you have a concentration gradient. Okay, for a normal cell, there is still some incentive for chlorides to go in. But there are exceptions where the concentrations are slightly different and the opposite thing happens, but I don't want to...
So it's hyperpolarization compared to the negative 60 to 70, or hyperpolarization to get it to the plus?
It depends. If it's at the resting potential, it will hyperpolarize it to the more negative. It's always to the more negative, okay? But sometimes it's hyperpolarization and sometimes it's just repolarization, basically, depending on what the resting potential is. No, okay. Okay, well, thanks.